So this research I'm talking about is research that's actually been done by a couple of my graduate students. The current one is Chase Nunez, and his contact information is listed there as well as my own. I might need a crash course on how to make, there we go. Okay, so we're going to start off with an introduction about anthrax. This is a bacteria, a soil-borne bacteria that essentially occurs worldwide. It seems to do better in alkaline soils, particularly calcified soils. And pattern that we see particularly here in Texas is a wet spring followed by a hot, dry summer. That kind of sets the stage for an outbreak. All mammals can be affected, but as we see, certain species tend to be more commonly affected. So in the soil, it exists as a very resistant spore form. This is probably of bacteria the most resistant bacteria known. So that spore really protects it from the environmental extremes. Once that spore gets introduced into the host, it develops into a vegetative or growing form that rapidly multiplies and eventually produces toxins, and it's the toxins that actually kill the host. When that host dies, those vegetative cells will actually die in the host if the animal is left intact. But what normally happens is a carnivore or a scavenger or a human or something comes and opens that carcass, and when that occurs, then those vegetative cells sporulate, become spores, and then again become very resistant. Anthrax has been around for a long time, folks. References to it in the Bible, probably the 5th and 6th plagues that you read about, may have played a role in the fall of the Roman Empire. So been around for a very long time. Let's see, is this the... There we are, okay. So in the United States, anthrax is more common in the western half. Again, kind of this calciferous soil band, so we can see outbreaks occur sporadically throughout kind of western United States and indeed throughout much of Texas. But we have what we call the endemic zone or enzootic anthrax zone, and this is this Edwards Plateau region. It's pretty well characterized, and we call that the endemic zone because that's an area that we see some level of anthrax virtually every year. Some years it'll be really bad, other years less so, but we'll have some cases of anthrax almost every year, whereas other parts of Texas and other parts of the United States, you may see outbreaks from time to time, but they're fairly sporadic. There are different manifestations of the disease anthrax. Certainly cutaneous, referring to the skin, that's what's most often found in humans because their skin gets infected with the bacteria. Humans can pick up gastrointestinal, same as animals. If you ate an animal that died of anthrax and didn't fully cook it, in fact, there have been cases here in the United States fairly recently of gastrointestinal anthrax. That's what we see most common in our animals, particularly in our herbivores. And then finally, inhalational anthrax. That's where you breathe it in. So from another animal that may have inhalational anthrax and they expire their air, this would be kind of analogous to the COVID. One animal expires their air and has the bacteria in those air, and you breathe that in, you can get it that way. That is also the way that weaponized anthrax is aimed at being distributed, so that you can release the anthrax spores into an environment from an aerial bomb or something of those natures, and people breathe it in. Very, very lethal. So fortunately, cutaneous anthrax is the least lethal, and inhalational is the most lethal, with gastrointestinal being somewhere in between. This is kind of what we see in nature, the cycle of anthrax. Okay, so this is showing a human as being our model of how they could get infected. So just going over again, they could ingest it from an animal that had anthrax. They can inhale it from the spores. This is showing those spores from the environment. That's actually very, very rare that you would get enough inhaled from the environment. Certainly, if you opened a carcass, it's conceivable you might inhale enough to get inhalational anthrax. But again, as we talked about, more likely in a biowarfare type of situation. Vegetative forms in the animal, again, those will die when that animal decomposes, but if that animal gets exposed to air, we get the spores that are highly resistant. Flies can play a big role in perpetuating anthrax. And again, you think about when do we see anthrax outbreaks? They're typically late summer, early fall. That's when we have a lot of flies. So you can have biting flies that can pick up anthrax from a carcass or from an animal that's recently infected and then bite another animal and produce the cutaneous form of anthrax. And what's not shown here in the picture is blowflies, the carcass flies, and the role that they can play, because they can feed on an animal that's died of anthrax. What those flies do is they gorge themselves, and then they go rest on vegetation, and they regurgitate. They basically barf up a bunch of the stuff that they just ate. Well, if what they ate had spores, now they've contaminated that vegetation with the spores that they picked up in the carcass. So there's a way that they can perpetuate the disease as well. Also, when an animal dies, and if the carcass is opened, those spores get released into the environment. Those spores can then contaminate the soil, but also any vegetation that's in the area where that animal died. So this is just a picture of a deer that died of anthrax and just showing the flies that are on it. Again, think about the seasonality of when this is occurring. Imagine all these flies potentially picking up spores and then going onto the grass or the brushy vegetation that surrounds this carcass and vomiting that stuff back up again, and what you've done to contaminate that environment, and remembering how incredibly resistant those spores are. So I just wanted to cover cutaneous anthrax, not because it's terribly important in terms of a wildlife situation, but certainly in terms of the human situation. The spore can enter through a wound, or as we mentioned, by a fly bite, and you get these lesions that are called an eschar. Okay? And what these are is they're painless. I mean, they don't look painless, but they are, in fact, actually painless. They're an ulcer with necrotic tissue in the center of them, so that tissue is dying off. Most cases, this is just a localized situation, a localized wound. It looks kind of ugly, but it resolves. Rarely, that bacteria can go systemic, can get into the bloodstream, and you can develop a bacteremia that releases toxins in the blood that can lead to enough toxic dose that you can die. That is fairly rare in cutaneous anthrax, but it can happen. And relatively recently, we had a case of cutaneous anthrax in Texas where it went systemic, and that individual needed he had to be hospitalized and needed some pretty intensive antibiotics to recover. Fortunately, he did. But so again, basically all mammals are susceptible to anthrax, but certainly the grazing species tend to be the ones that we see the most cases in. Obviously, our wildlife species are deer and other grazing herbivores, and then our livestock as well. Some of these animals will die incredibly quickly. I've heard people talk about watching a deer walking down a trail and just literally keeling over. There may be a little bit of exaggeration in that story, but certainly these animals can die very quickly once they get infected. So that's a paracute. So again, you may see an animal that's staggering and then collapses and keels over and dies. You may or may not see some convulsions before that. That's a fairly rare paracute, meaning occurring within less than a day. What we'll typically see, particularly in livestock, are more the acute cases. So this is cases that, again, they die pretty quick, but it's a couple of days. So these are cases that, with livestock at least, if you can get your hands on them fairly quickly and get them treated, you might have a chance of turning them around. Abrupt fevers, depression, stupor, respiratory and cardiac distress, staggering, and then some things that go along with that. Anytime you really stress an animal out, abortions can occur in pregnant animals, so that's not unusual. The hallmark of anthrax, what everybody always thinks of when you talk about anthrax, is bloody discharges from orifices. In fact, some people will say, well, if you see that, that means that the animal died of anthrax. Well, I have seen lots of animals with bloody discharges, particularly after they're dead. Actually, only a minority of those were anthrax. If the animal dies, some other things you can look for. Absent or incomplete rigor mortis, okay? And failure of blood to clot. Now, rigor mortis can be tough to judge sometimes because that depends on how long the animal's been dead as to whether it would have set in or not. But blood should normally clot. So that's one of the reasons why you'll see blood oozing from orifices, because it's failing to clot. Hemorrhages, again, hemorrhages in the body cavities. Obviously, EHD is one thing that can cause that, but there are other diseases as well. Enlarged organs, one of the most typical ones is what they call the raspberry jam. I don't like that term because I like to eat raspberry jam. But the spleen basically turns to kind of a jam-like consistency. It's very dark and semi-fluid as opposed to its normal consistency. But other organs as well can be enlarged, including lymph nodes. And again, lymph nodes can be enlarged from a variety of different things. So don't think just because you see enlarged lymph nodes it's got to be anthrax, because it certainly could be other things as well. For domestic livestock, in endemic areas, this disease is pretty well managed because we know we can vaccinate animals pretty effectively. There's the Stern strain of vaccine produced by Colorado Serum Company. That Stern strain of vaccine was created by Max Stern. Actually, it wasn't created, it was actually discovered by Max Stern in the 1930s. So it's been in use for almost 90 years. A very safe and effective vaccine with a couple of limitations. First of all, for best effect, you need a booster. You vaccinate, and then you need a booster 2 to 3 weeks later. And the immunity that's produced is quite good, but it's short-lived. So that immunity will only last 6 months, maybe less. So ideally, you want to be vaccinating, get them fully vaccinated 2 to 3 weeks before you anticipate the anthrax season coming on. And with that booster, that means you're going to look at your first vaccine's got to happen at least a month, but hopefully not more than 3 months before you start seeing cases. But again, in these endemic areas, ranchers are very accustomed to dealing with this, and they've got this on their schedule of how they're going to vaccinate. When it comes to wildlife, that's not so easy. Pretty hard to get our hands on wildlife and give them a subcutaneous vaccine. As I mentioned, if you have an outbreak, livestock, if you can get them gathered up quickly, you can treat them with antibiotics, it can be effective. It's not going to happen with wildlife, that's impossible. So we're usually looking at trying to manage this in terms of a wildlife situation, trying to manage the outbreak to prevent it from getting any worse. And the best thing we can do in the face of an outbreak is to minimize the number of carcasses that are out there, get them disposed of quickly and appropriately. Again, that's easier said than done. It takes manual hours and labor, and there's some human health risk that goes along with collecting those carcasses. This is the recommended thing to do, is to burn the carcasses, because the burning will destroy the spores if those carcasses are fully incinerated. That's why they've got the tires here. Well, when do we have anthrax outbreaks? Where do we have anthrax outbreaks? Is it a good idea to be setting things on fire in those situations? Probably not. I mean, you just look at this vegetation that's surrounding this carcass, and you'd better have somebody there with a bunch of water to be monitoring that thing, or else things are going to get out of hand. The other alternative is deep burial, burying them like 6 feet deeper or deeper. When you think about our anthrax zone, what kind of soil or lack thereof it exists in that area, deep burial is also not a very user-friendly situation. "2019 is the most recent bad outbreak that we've had of anthrax in the state of Texas, at least in terms of numbers. And I had a map that's showing this outbreak, but that map didn't work out very well for whatever reason. But if you think back on the map I showed you, this outbreak occurred in the historic endemic area, in the Edwards Plateau. So in terms of geographic range, it was not that unusual, but it was definitely unusual in terms of the number of animals that were lost. Had a couple of unaffected counties that we might normally see, but our losses were estimated in terms of wildlife losses were estimated to be 10,000 or more. That's a lot of animals that died in that anthrax outbreak. This was a ranch that I worked with during that outbreak, and this was his bone pile. He had a lot of exotics on the ranch, and so you can appreciate this guy lost a lot of money in this outbreak. So that was 2019. That was our big anthrax outbreak and kind of a historic event. But it was followed by a couple other historic events in 2020 and 2021. Outbreaks in the panhandle of Texas. Well, again, that lies within the historic anthrax area, but we haven't seen outbreaks there in 50 years. You think anybody's practicing livestock vaccination in an area where they haven't had an outbreak in 50 years? No, especially when you consider when you would have to be vaccinating those animals, not when you're normally going to be gathering them anyways. So no, in fact, they had not. In fact, I got calls, the way I first found out about it was getting calls from veterinarians in that area wanting to know if our oral anthrax vaccine was up and ready for use because they were wanting to use it in cattle. Not in wildlife, but in cattle because they had cattle out on the range in some pretty rough country where it was going to be impossible to try to gather those things to vaccinate them. Well, I told them we're not even close to having this vaccine ready for deer yet, and cattle, would be a whole different ballgame. So sorry, we really can't help you, but bad situation. These outbreaks were fairly localized, but had significant mortalities on those ranches that were affected. And the animals that were lost were primarily cattle and deer, and a few horses. Interesting, at least one of the ranches also had quite a few aoudad and did not lose any aoudad, which was, I don't know why. So this is showing those, so again, 2019, our historic anthrax area, not a big deal, but in 2020, we're having the outbreak here just south of Amarillo. And the following year, kind of the same areas in the Panhandle, but some different counties involved. So it kind of just goes to show that we can have anthrax outbreaks occur anywhere the soil conditions are right, and sometimes with very long gaps in between. One of the problems that we see as anthrax researchers, particularly when we're trying to get funding for development of this vaccine, is the USDA says, this is not a serious disease because there's hardly any cases that ever get reported. Well, that's true, and that is an issue that you deal with all diseases, with all livestock diseases. People don't like to report, or some people don't like to report the fact that they've got a serious disease on their place. They don't want the government to know. They don't want to be put under quarantine, those kind of issues. And with anthrax, it's even worse because there are requirements that if you had an anthrax outbreak on your property, when you go to sell your property, you've got to reveal that to the buyer. The reality is it doesn't make a dang bit of difference. If you're in the anthrax zone, whether your property has had an outbreak recently or not, doesn't mean that it's at higher risk than any other property. They're all at risk. In fact, as we've seen in the Panhandle, even though you haven't had an outbreak at 50 years, you're still at risk. So that doesn't mean anything. But when you think from the perspective of an out-of-state buyer who doesn't understand anthrax in Texas, and the people who pay a lot of money for ranches a lot of times are out-of-state buyers. And when they hear there's anthrax, oh my God, I'm not buying that place. I'll buy the place right next door. Okay, well, yeah, you saved yourself. So again, when we talk about 2019, the estimates are 10,000 animals, wild animals died, wild and exotic. But we don't have firm numbers on that by any means. So one of the things that we're trying to do is we're trying to estimate wildlife exposure to understand the extent of the problem and also to help with funding. And so we are looking at, well, we know we can't really rely on the numbers that are reported because of underreporting. So what are there other things that we can do? Well, one of the things that we're looking at doing, and this is one of Chase's projects, is to document levels of exposure by looking at animals that have survived anthrax and have antibodies in their blood that we can detect to create a heat map of what our anthrax prevalence is in the state of Texas. And so there's his phone number if you want to contact him. He has already collected about 175 white-tailed deer blood samples and 75 or so feral pigs. We're interested in whitetail because that's our primary species of interest in terms of the management, but feral pigs because pigs are actually fairly resistant to anthrax. So they're more likely to survive, which if we're wanting to see if an animal's been exposed, you need that animal to have survived. So that's one of the reasons that we're interested in looking at pigs. So if you have properties in or near these endemic areas or any of the outbreaks and would like to participate, please contact Chase. At this point in time, this would be my recommendations. And these recommendations were really geared towards captive or exotic producers. But anytime you have a wild animal in your hands, if you are in the endemic area, I would recommend that you vaccinate them with the Stern strain. As I said, the immunity that results is fairly short-lived, and so if you're in the wrong time of year, you may say, well, maybe I shouldn't be vaccinating just yet. It's not the right time. Well, you just don't get your hands on these animals that often. So if you have your hands on them, the vaccine is cheap, you should do it. Or I was at a capture a couple of years ago where they were moving a bunch of animals to the anthrax endemic zone and didn't think about, should have put some vaccine in those animals. I was not the veterinarian in charge, so it wasn't my fault. I wasn't the one that overlooked it. But I was talking to the veterinarian and said, yeah, you know, you probably should have given those guys a dose of the Stern strain before you loaded them in the trailer. Yeah. The other thing that we've had people do is attempt to vaccinate animals with the vaccine by darting them. If done properly, this can work. The problem is you have to get ahead of it. So our lab has validated, at least in terms of developing an immune response, if you basically follow the label directions, but you deliver the vaccine by dart, at least in our hands, we were able to get an immune response in white-tailed deer. Now, those studies were not challenge studies. In other words, we didn't subsequently expose those deer to anthrax to know that they survived, but based on their immunity that developed, we would expect that they would. And then, of course, hopefully in the future, we'll have an oral vaccine, a vaccine that can be put into a feed bait of some kind that animals can consume, and you can pick up the vaccine that way and get protected. This isn't the first time this has been tried. In fact, when I was a kid, I got polio vaccines in a sugar cube. Anybody else get vaccinated that way? Yeah. And we actually vaccinate against rabies in coyotes and foxes and raccoons back east in an oral vaccine. Okay? So that works well. And there's some other oral vaccines that have been used as well. So that's what we're looking at kind of long-term down the road. Dr. Jamie Ben Felix was my previous PhD student who looked at this and developed this in mice. So how many of you are wanting to protect the mice on your property from anthrax? I didn't think there's very many of you. But mice are pretty cheap and a good place to start in the lab, and so that's what we did. And how we did this was microencapsulating the Stern vaccine. And so that means basically putting the vaccine in really small balls, really small spheres. And so the concept is a gumball and a gumball machine, and where the gumball is the spore vaccine, and the gumball machine is that sphere, that microsphere that the vaccine is in. And the idea is it's going to stabilize that vaccine when the animal eats it, stabilize it in the bait, and stabilize it in the animal's gastrointestinal tract so that it makes it through the entire GI tract to the end in the small intestine and the large intestine, where there are lymphoid follicles that can pick the vaccine up and then distribute it through the blood as would have happened in a subcutaneous vaccine. I'm not anywhere near smart enough to develop this technology to do this kind of stuff. It uses a lot of biochemistry. I'm not much of a biochemist. In fact, biochemistry almost kept me out of vet school. So I don't try to use it today. But Dr. Allison Fick, she is a biochemist. And so she's the one who developed this vaccine and developed it for brucellosis originally, using the same concept. And so we just substituted the brucellosis vaccine and put in the anthrax vaccine. We thought this was going to be really easy. We forgot about the fact that Bacillus anthracis, the anthrax bacteria, does this alternation between vegetative and spore forms of the bacteria. Brucella doesn't do that. So Brucella is a lot easier. That sporulation makes the vaccine development for anthrax quite a bit more complicated. But Dr. Ben Felix worked through that and got it to work. One of the things we've heard anecdotally is that some ranchers would take the anthrax vaccine and pour it over corn, for example, that they had deer coming into, and they said, well, we dump it over corn and we'll protect the deer that way. It wasn't a bad idea necessarily, but at least when we tried that in mice, it didn't work. There was no immune response that developed. But when we gave them the encapsulated vaccine, the mice did produce an immune response, and an immune response significant enough that it could neutralize the anthrax toxin. Again, we did not try to give these mice actual anthrax disease, so we didn't challenge them, but based on their immune response, we would expect that they would be able to handle an anthrax exposure. And that's what this is showing here. So this is the vaccine. Saponin is just the standard adjuvant that's in the vaccine. So this is just the vaccine given to them orally straight, no response. And here's the vaccine given to them in the protein shell microencapsulation. And you see we got this response. And I think that's reading the 50% line. That's where we expect protection to exist. But obviously, we're not interested in protecting mice. We're interested in protecting deer and other species. So this is Chase Nunez here, working on his PhD project, still evaluating these results, but it's looking promising. So initially what we did was we worked at Texas A&M has a small wildlife center with a small cadre of deer. I think there's 12 or 15, 16 deer, something like that. And we tried different formulations of the vaccine and different concentrations. And so this is looking at, here's deer that were given the subcutaneous vaccine the way it normally would be delivered. And you see this pretty dramatic increase in titers. But again, you can appreciate how that titer drops after about 5 months. We're down to not very much. And here's our oral vaccine, and so it wasn't as robust by any means as the injectable, but we did get up above that 50% there absorbance, which relates to having toxic neutralization aspects, which should prevent the disease from going on. So this was this kind of pilot project that was done at the Wildlife Center. This is Dr. Alice Blue, who manages that center. And then from that we've now conducted a more extensive project using 36 deer total, using the Flying Bee Ranch and Texas A&M Kingsville, the Caesar Clayburgh Wildlife Research deer, so we could get larger numbers of animals vaccinated and look for responses. So that project is now completed in terms of the animal handling and the animal vaccinations and blood draws that came subsequent to that, but we're still analyzing the results. So this is kind of, as Jacob said, stay tuned. It's kind of a stay tuned situation for you. We're also interested in trying this in other species besides white-tailed deer. White-tailed are our primary focus, but we recognize that certainly in the exotic industry, there's some more valuable animals that may be out there that these producers might be interested in seeing protected. And so we've got a cooperator who's got some axis deer that he was interested in trying this in. And so that's what we're doing. Again, when darting animals with the vaccine, as I mentioned, we've validated that in white-tailed deer, but in other species we've had some exotic producers that have had some luck doing that and others who didn't. I suspect the ones who didn't, it probably was a timing situation. They didn't get them vaccinated quickly enough before the anthrax exposed them, so they didn't have time to achieve an immune response. But we're in the middle of this project right now where we're hand injecting some of the axis deer, darting some with the vaccine, or using our oral formulation to compare and see what their immune responses are. And we're about halfway through this project, got a couple of months to go still. So I do want to recognize the funders that have occurred so far. Certainly A&M's College of Veterinary Medicine provided probably the bulk of the funding for this so far. Center for Disease Control, the Exotic Wildlife Association, the Second Arc Foundation, and we've had a private contributor who gave us some funding as well. And appreciate the collaborators that have worked with us, the Winnie Carter Wildlife Center there at A&M, Flying Bee Ranch in Seguin, and Caesar Clayburgh Wildlife Research Institute for white-tailed deer, and then where we're currently working at the Diamond R Ranch for the axis deer. And with that, I can take any questions.